نعطي محاضرة اليوم عن بسم الله الرحمن الرحيم عن regulatory T cell functions. راح تكون محاضرة should be short and really depends on the previous lecture, so it should be a short, easy lecture today. So regulatory function of the T cells. So many of the things today we know are just continuation of previous things that were discussed. So the T cell is the main player in the regulation of the adaptive immune response in both its arms, as we know. So helper T cells are required to directly activate specific B cells to differentiate into plasma cells, as we know. So to produce antibodies to certain atypical antigens, مثلا large molecules, مثلا capsular polysaccharides that are multivalent, they can attach to antibodies at more than one side and can cross-link them. So such as antigens along with haptens مع اختلاف بسيط, you would need a T cell independent production of antibodies. So micromolecules, polysaccharides, DNA, RNA, أشياء يعني a molecule that is made up of multiple molecules that are very similar. مثلا DNA, CG, CGT, AT ورا بعض كتير يعني very similar. Polysaccharide actually the same saccharide with over and over. So these are repeating subunits, and obviously they can attach multiple antibodies and they can cross-link them. So the repeating nature of their structure multivalent, they cross-link the IgM. الIgM هو أصلا هو لما يطلع بكون pentamer. تذكروا حكينا إنه it'll be a pentamer, and this antigen receptor on the B cell surface activates it. شو بصير؟ So عفوا pentamer هذا بالبلازما, but it's a monomer on the cell surface of the B cell. فبما إنه هاي الموليكيول راح تربط أكثر من IgM على السيرفس تبعت ال B cell, they will cross link in the same manner اللي حكينا الكروس لينكينج بصير بالمثلا ال IgE bound مثلا cells لما يصير مثلا cross linking بالهابتن حكينا. That's the same thing. So because this DNA molecule is large and very similar, or polysaccharide, they will bind to many IgM surfaces on IgM receptors on the surface of the B cell. راح يعملوا this clump, cross-linking clump of receptors جنب بعض, and this will activate the B cell without the need of a helper T cell. These large macromolecules هم بنسميهم هذول بيعملوا لك antibody, the T cell independent production of antibodies. So the T cell dependent antibody production, this is the direct activation اللي احنا بنعرفه اللي هو that we know. So you have an activated T cell تروح على B cell, and then you have the CD28 من ال activated T cell with the CD40 ligand. هذا بنحكي هذا كله. So you have this activation. This is the direct activation into, and in the end I want to make a plasma cell and a memory cell and clonal proliferation of this cell. So what happens? So in this direct method, the antigen is processed by the APC, and then I present it to the helper T cell, and then interaction between the T cell receptor will MHC2, and then the cell is then activated, and then following its interaction with IgM RGD on the cell surface of the B cell. So once the antigen cellular internalization within the B cell, the epitope is linked to the MHC2 زي ما بنعرف, presented to the APC. The APC will then the interaction راح يصير بين الهيلبر T cell وال B cell, and then we'll have a co-stimulatory signal given by اللي هو ال B7 protein with the CD28 مع ال T cell, and the helper T cell is then stimulated to produce interleukin 4 and 5 as we know, and 2 to produce itself, to activate itself. This production اللي هو dependent antibody production will class switch from IgM who the first class of immunoglobulins in the acute phase to the chronic phase اللي هو IgG, IgA and IgE. And then so this is what we're saying here. The antigen is produced on the B cell, راحت على T cell, presented it, interleukins were produced, this T cell is now is active, it activates the B cell, بتروح تعمل memory أو تعمل plasma cell, and this is the activation between the اللي هو the MHC2 and then the activated T cell receptor مع CD28 and CD40 مع CD40 ligand as well. So the CD40 comes from the B cell and the CD28 comes from the T cell, and then they all three together will activate the B cell. So تمام. So there's the third signal اللي حكيناها. This is similar to the co-activation with B7 CD28 with the fully activated اللي هو CD40 ligand. This protein interacts with the resting B cell, which helps them differentiate into plasma cells. So other proteins on the surface. So this is the third signal between now fully activated T cell, the CD4 ligand, as we mentioned, the CD40, which helps them differentiate into antibody producing plasma cells. So other proteins on the surface of B and T cells to strengthen. They only strengthen, يعني بقوة الانتراكشن between these two cells. هلا في نقاط كثير مهمة نعرفها. الأولى يعني ممكن نستنتج إنه a T cell dependent response, all classes of antibodies are produced at first من البداية. So if you don't have to wait to class switch زي ما عملنا بالindependent response. إنه I want to find a hapten or a thing and conjugate it to a protein or أروح ألاقي cell و get my activation license, right? I don't have to do that. I get the license from the beginning. So T cell independent حكينا primarily is IgM produced وأسرع, right? So that really explains why in the beginning of an infection عادة IgM هو البداية. It stays for a few weeks, couple weeks, and then you start IgG becomes the dominant antibody. هذا الأول. ثاني, only the T cell dependent response اللي بصير فيها to activation of T cells will produce memory B cells. An independent response keeps no memory. لكن a T cell independent response will react faster. So it is a T cell independent response أقرب إلى خلينا نحكي إذا على السبكترم هو أقرب إلى innate immunity من ما هو adaptive immunity. هو still fully adaptive immunity, but it has some shares few similarities with some elements of the innate system. So the T cell independent response is the main response for bacterial capsular polysaccharides, DNA, RNA زي ما حكينا. And these molecules are not processed and presented by APCs, and obviously they do not activate T cells. So T cell independent response. So this is in part similar to the hapten hypersensitivity زي ما حكينا. It bypasses the need for the T cells. It can activate non-peptides. It produces no memory. So these molecules are not usually small, but are large molecules, non-peptide based. They can cross-link the IgM on the B cell surface, and they cause full activation and antibody production without the need of presentation to the CD4 cells. So two types of antigens produce a T cell independent response. عادة ال B cell mitogen اللي هم these هذولا are molecules that cause a proliferation of B cells, mitogens. So it will produce obviously polyclonal sometimes molecular low concentration. عادة أكثر شيء polyclonal. إيش يعني polyclonal؟ يعني I have different clones of the same antibody. So all the antibody reacts to the same antigen, but كل واحد له شكل مختلف. يعني مش 100% مش متطابقين 100% زي المونوكلونال. So these are polyclonal. They will produce varying response. It still produces a response, but من قوي لضعيف وهكذا. So the activation occurs due to the reaction between these molecules and toll-like receptors on these B cells. So هيك شفنا إحنا now the link between the innate immunity. قاعدين نحكي عن toll-like receptors. So the activation occurs due to the reaction between these molecules, and I have toll-like receptors on the B cells. Now the B cells are activated by these mitogens that activates them without the need for T cells. So bacterial DNA, LPS, endotoxins are هدول هم كلهم مش بيبتايدز وكلهم هدول are B cell mitogens. الثانيين هم اللي حكيناهم بالسابق اللي هم large macromolecules that cross-link a large number of IgM on the B cell surface. So these are the two kinds of antigens that produce a T cell independent response. So here نقطة ثانية عندنا إنه so immature B cells, those in young children, produce energy response when you try to activate them with large macromolecules. That's why unconjugated vaccines do not elicit a response in these children. So we conjugate them to proteins so we can produce a dependent response instead. Bacteria capsules are the classic examples. DNA, RNA molecule, macromolecules. يعني حكينا molecule متشابهة من قطع كثير متشابهة. So why do we conjugate vaccines? We conjugate a weak immunogen capsule مثلا to a non-strong immunogen مثلا typhoid toxoid. So you will produce a high response against a weak antigen. An antigen otherwise would produce a weaker response. Children عندهم less mature IgM on their cell first, so we need to help them, including the T cell independent loop, to present antigens to the T cells to help them activate the B cells faster. I think this one. I think this one is clear. So will there be any T cell dependent activation here? Think about it. Can it be responsible for any side effects in a vaccine or improve its efficacy? So the answer would be yes. Okay, cell-mediated immunity. So we have a reminder here that the regulatory function of the cell-mediated immunity is similar to the direct T-cell dependent activation. So I think we have covered this. We have covered this before in a previous lecture. And we were talking about CTLA4 and CD28 positive and negative reactions or co-stimulatory signals. Suppression of certain immune system of immune responses. مثلا في عندي أنا regulatory T cells subset منهم نسميهم they are inhibitors for the effector functions of T cells or CD4 or CD8 cells. These are called suppressor T cells. هذول are about 5 to 10% of CD4 positive cells and characterized by having CD25 as a marker. They produce a protein that regulates the transcription of other various genes. هذا مثلا اسمه واحد منهم اسمه FOXP3. هذا هو inhibitory factor which will produce CTLA4 حكينا عنه سابقا. So this is the inhibitory way of how these cells will initiate the production of CTLA4. هلا الناس اللي ما عندهم FOXP3 الجين, so obviously they will not be able to produce CTLA4. هذول بيكونوا predisposed to autoimmune disease مثلا SLE and other rare diseases مثلا polyendocrinopathy and enteropathy as well. So this is from the previous lecture, and this is where FOXP3 controls this portion. Regulation of T cells is extremely important, as we know. So مثلا a person with leprosy, they have uncontrolled multiplication of Mycobacterium leprae. This is an intracellular organism, much like tuberculosis. It needs cellular immune response. So a lag of delayed typhoid hypersensitivity, the immune response, the proper one, will lead to uncontrolled growth and excess of lesions. So you have a lot of CD8 cells that are not doing anything in these lesions, قاعدين بيحاولوا يشتغلوا, and then they are not able to do so. So removal of some of the CD8 cells can restore cellular immunity in such patients and limit the multiplication of this organism. Another example, normal individuals, whether they have a ratio of CD4 to CD8 about 1.5. People with AIDS راح يصير الخلية that is affected is CD4. So CD4 keeps coming down. The lower it goes, the more new infections start to come up in this patient. So the normal ratio will start to get reduced أقل وأقل. لما يكون مثلا CD4 count is below 200, this is very dangerous and lots of infections start to occur. So obviously CD8 number cells will increase, creating further imbalance in this ratio as well. Third example, so an important mechanism of مثلا host response to infection is the increased expression of class 1 and class 2 MHC proteins. So if you have more MHC proteins, so more antigen presentation, more detection of مثلا intracellular antigens. So some viruses, they suppress this MHC1. Why? We know why, because they're trying to mask their presence inside the cells. B cells perform two important functions. We know them. Plasma cells, they are also antigen presenters. So it's important to know that an activated B cell requires antigen. It's antigen dependent to be activated, whereas an immature one is not antigen dependent. It keeps maturing its surface receptor. So between mature to activated, I need an antigen. Otherwise, it keeps just maturing on its own. So pre-B cells differentiate into immature B cells without the need of antigen presentation. They even mature to naive mature B cells without the need of antigen presentation. So they do need certain proteins for the pre-B cells to differentiate into the immature B cells. هدول اسم بسموهم protein tyrosine kinase. It is a signal transduction protein. If this is mutated, this is an X-linked, بسموها X-linked gamma globulinemia. So they have no production of gamma globulins اللي هم antibodies. So if you don't have this protein kinase, which is the signal required to keep going through here, all the way لحد هون. If you don't have it, then you will never have mature B cells, and you will never be able to activate B cells and then produce antibodies. So they have a gamma globulinemia, they don't have antibodies. Obviously, they will have severe infections, pyogenic bacteria. Again, I need antibodies to clear these capsulated bacteria as well. So this surface IgM is a monomer, one molecule. In contrast, when it is secreted, it is a pentamer. When زي ما حكينا, it's too large to cross the placenta, and the baby is usually protected by IgG, and then حكينا IgA from the milk. The monomeric IgM on the surface has an extra transmembrane domain that anchors the protein. So basically نفكر فيه إنه IgM مع قطعة إنها تلزقها على هذا. Surface IgD may also have a similar thing. Pre-B cells are found in the bone marrow, we know this. The mature lymphocytes have relatively short lifespan, and this is compensated by high production of lymphocytes each day. Within lymph nodes, they are located in the germinal centers, and within the spleen, they are found in the white pulp. The white pulp, red pulp, تعرفوا, right? White pulp is where all the lymphocytes will be present. In the gut, it's in the gut-associated lymphoid tissue, GALTs, مثلا مثل Pyrus patches. So clonal selection, how are antibodies made? Selective. رح ناخذ محاضرة عن antibodies لحالها بس. So the current theory, which is most accepted, is that what we covered حاليا, إنه an antigen is basically wait to be picked up by a B cell that has already selected the variability to this receptor, and this is clonal selection. So each individual has a very large pool of B lymphocytes, each immunologically responsive to a surface receptor that can react to only one antigen, and it's estimated you have at least 10 million different specificities. وفي حكيت لكم إنه there is a antibody library that في جامعة معينة has this library of all different specificity of different antibodies in humans. إلى الآن وصلوا إلى مليار different antibodies, each reacts to a different antigen. So basically this is a key lock mechanism اللي هو antibody antigen with B lymphocytes. It has the lock and looks for the best key for this lock. So once these B cells are selected, they become plasma cells, as we know, and all the other mechanisms that we mentioned. So we will talk later how they have the same. Okay, so in immunoglobulins, بالantibodies, they all have the same. حكينا there's a constant part and a non-constant part, so they all have the same heavy chain and the same light chain as those carried by the selected B cell. And when you class switch from IgM to IgG, only the heavy chain is changed. متقبلي light chain موجودين. So this is how you changed the class. We will cover this in the lecture about antibodies. The process of activation of B cells. So this will occur in the following manner. مثلا عندك multivalent antigen, it binds to the surface IgM مثلا and cross-link مثلا critical number of these surface antigens. This immunoglobulin aggregate will become into a clump, form a patch on the pole of the cell. Endocytosis of this cap of the immunoglobulin material will ensue. هذا الانتيجين كله with the immunoglobulin is then processed وبتطلعوا الابيتوب زي ما حكينا. When you digest these proteins, you find the most unique parts, and then they are presented on MHC2 molecules. So and then the story goes on as we know it. Now I need the two similar signals, the CD28, the CD40, 40 on the T cell with the ligand عفوا مع CD40 on the B cell and 28 on the T cell مع B7 on the B cell. So this will prevent allergy, and then this also is required to class switch. So there's a syndrome called hyper IgM syndrome. إذا ما عندهم CD40 ligand, they cannot class switch, and they always have IgM and no other antibodies, and they have severe pyogenic infections as well. And this is what we cover in just now. What are the effector functions of B cells? Basically produce a large amount of immunoglobulins. كل واحد مختلف. It's the same one as this surface antibody can, only specific to one antigen. It will produce the same specific antibody. Basically, just remove the part that makes it a surface antibody, and then keep producing a very large amount of very identical antibodies. Call them the same protein, basically, over and over and over and over. Most memory B cells have surface immunoglobulin that serves as this receptor. Some have IgM. تمام. So memory T cells secrete interleukins that enhance antibody production by the memory B cells. Okay, and we will stop here. ويعطيكم العافية.